Are we in?
I think so. Hello, everyone. I hope that you can see us. We are very excited to have Marina Bedny today. As before John introduces Marina, as a reminder, the Learning Salon is a forum where we discuss interdisciplinary bridges and contentions between artificial and biological learning. We have an interdisciplinary audience from different stages in academia. So if you have any clarification questions, feel free to, during the talk, add your questions in the chat, and we all share resources and answer each other's questions. We make sure to be very respectful. And hi, Steve. And also talk to each other respectfully, but disagreement is totally normal here because our goal is understanding, and disagreement is never a sign of disrespect unless it's mentioned disrespectfully, in which case we step in if people talk over each other or interrupt, or we try to make sure that we manage the atmosphere in a way that it feels conducive to constructive conversations. If you have any disciplinary differences or questions, or you feel too junior, don't worry about it. Ask your questions anyway, because there would be probably other people in the audience who feel similarly to you. There is a ask a question button below, and you can press that and type in your questions and vote on each other's questions. If you don't want to be asked to join the screen, please say ask for me. Otherwise, we might ask you if you're around to join us on the screen. With that, I hope that everyone is starting the year joyfully. It's our first session of 2022. I cannot believe this. We start in 2020. And yeah, it's a joy to spend this extended pandemic with all of this virtual community. And yeah, with that, I would like to pass it on to John.
Ida, Ida, were you not gonna say something about you mentioned
Nature.
something? Yeah, I don't actually know what that is, so I'm being self-serving. I wanted you to I've been in isolation and quarantine, so the whole world is just unknown to me for the last week. So what's been going on? Ida? Oh, her mic doesn't work. Well, while Ida's trying to get her mic to work, I'm going to introduce you, Marina. You can hear me, right?
Okay. Yes.
Yeah. So it's really very exciting to introduce Marina Bedny today. Now, of course, Marina is a colleague of mine at Johns Hopkins, so which means I never met her at Johns Hopkins. We actually met online on a joint study section at the NSF. And that's what always happens, right? You meet colleagues who live next door to you when you go to meetings, either remotely or for real. And Marina does extremely deep work that is one of those scientists. She's one of those scientists where if I had my time again, I would want to do that kind of work myself. It's just very conceptually juicy and very provocative, and it's in an area that I also love very much. But what is cortical plasticity, and is it really different when you're young developmentally versus later in life? Just to give you a flavor, Marina does this really fascinating work in congenitally blind versus late onset blindness and makes distinctions between them, and asks the kind of questions that you would not initially think one should ask, but actually they take you into very, very deep conceptual directions. So it's extremely exciting. And without further ado, Marina.
Thank you, John. Can you hear me okay?
Yep.
Thank you for that wonderful invitation. Your invitation is a hard act to follow, but here it goes. That's lovely. So I'm going to share my screen now. Is that how I should start? Okay. Let's see.
Testing. My mic works now, right?
Works perfectly for me. Perfect. All right. Can you guys see my screen?
Yes.
Okay, fantastic. I'm gonna try and keep a track of the time because otherwise I will just talk on and on. Thank you for inviting me. It's an honor to be here. I'm really excited to talk with you today. And what I want to talk to you about is how the human brain adapts to the varying niche that we deal with as humans. And I'll explain what I mean by that in a second. Um, and what I'm going to do in this talk, since it's a short amount of time, but I'm still going to try to give you um, a couple of examples of the way I think the human brain does that. The work I'll talk about today is going to focus on cortical plasticity, studies of cortical plasticity. We also do work that it will be another time and place fun to talk about, which is how people learn about the world from language as opposed to from their sensory experience. But I don't have time to talk to you about everything today. And I really wanted to get to this work by a very talented graduate student of mine, Yunfei Liu, on programming. So that's what we're going to do. So what are the so the question of interest that I'm particularly interested in are these two interrelated questions. So one is, how does experience contribute to the mind and brain? So this is a nature nurture question. How, to what degree or in what way are our mental and neural capacities built into our brain, and in what way do we learn from our environment? So that's one flip side of the question. Another side of the question is, how does the brain adapt to our varying niche? And these two questions stem from kind of this duality of what I think it is to be human. One is that we're these incredible creatures that are particular. We're different from other animals in our intelligence, right? In the cognitive abilities that we have. At least we think so. We do things that other animals don't do. We have language, we do mathematics, we have social structures like governments, we do all sorts of amazing things that are not done by other animals. And one way we might do that is by specialized neural and cognitive adaptations. On the other hand, another characteristic of humans is just the variety of ways that we live, right? So most animals are confined to a much narrower niche, whereas humans do all sorts of things and vary and thrive in environments as varied as New York City and the Antarctic, and adapt to things like sensory loss, learn cultural things like coding expertise. So what I'm going to try to think about with you today is, how do these two things work together? How is it that we both seem to be highly specialized and able to achieve incredible cognitive feats through that specialization, but yet highly adaptive? And so the issue that I want to talk with you about today, or one starting point that I want to take, is this question of how our brain adapts to cultural variation or experiential variation. And I think, the best work on this topic comes from DeHaan and Cohen in their seminal paper called Cultural Recycling of Cortical Maps, which proposes one way to think about how our brains adapt to things that they didn't evolve for. And in the two cases that they take is reading and mathematics. And what I'm going to do with you today is take two other cases, and we could return to reading actually and math if we wanted to discuss that at the end, to try to think about this problem with them. So one idea that they propose in that paper is that the way we adapt to cultural specialization is by recycling of domain-specific cortical maps. So cortical map that's built for, object recognition of a particular variety. So for example, recognizing line junctions gets tweaked for the purpose of reading. right? And we form representations of letters. And what I'm going to argue today is that actually that's not the only way that our brain adapts. That on the one hand, I'm going to show you evidence from blindness that the brain is much more plastic than that. It's not just the case that we can take maps that we are born with and slightly tweak them. We can overwrite them with new maps early in development. So that's going to be the blindness part. And then I'm going to turn to a completely different example, which is coding expertise, and to suggest that one major way that we adapt culturally, in particular for cultural symbol systems like coding, maybe also aspects of mathematics and formal logic, is by using domain-specific reasoning mechanisms. But in this case, I think it is kind of like adapting pre-existing cortical maps. So let me flesh it out. So the first topic I want to talk to you about is plasticity in blindness. Blindness has been of interest to psychologists and cognitive neuroscientists and philosophers for a long time. George Berkeley wrote about blindness as a way of thinking about how experience shapes the mind and brain. And what we've learned by working with people who are blind is that actually, contrary to some early ideas that philosophers had, people who are blind have mental representations of the world that are in many ways very similar to those of sighted people, and know all sorts of things that people used to think are visual ideas, like, about how color works and how light works. But there's one part of the brain that changes dramatically in blindness, and that is the visual cortex. So this is this large part of the brain that we believe evolved for visual perception, for recognition of contours and color and spatial perception. And so one thing we can do by working with people who are born blind is to ask, to what degree can this part of the brain change in a person who from birth doesn't get any visual experience? And the general answer to that question has been that there's a potential for change, but actually not very much. So the idea being is that, a long time ago, people thought that maybe this part of the brain would atrophy in a person who's blind. We've known for 30 years that doesn't happen, that this part of the brain takes on other functions in people who are born blind. But the received wisdom has been that those functions are analogous to vision. They're just in a different modality. So for example, you might do spatial localization in audition, or fine-grained spatial discrimination, but via touch. So the part of the brain, it maintains its usual spatial localization kinds of functions, but does it over information in different modalities. And what I'm going to try and tell you today is that we find evidence for something quite different, which is that this visual part of the brain in people who are born blind takes on higher cognitive functions, including language, and becomes sensitive to syntax, and also the difficulty of math equations. And what I think is happening is that higher order cognitive networks, including language networks and frontal parietal numerical cognition networks, invade the visual system and carve it up and take over it. And we can talk about why that happens. But first, let me tell you the evidence. So the first evidence I'm going to tell you about is from people who are born blind. So these are individuals who have at most minimal light perception. Some of these participants don't have any light perception due to some pathology with the eyes or optic nerve, so not due to brain damage. These are individuals who are matched to the sighted participants on age and education, and they have no cognitive or neurological disabilities, and they perform, like the sighted participants, standardized tasks where we can measure things like vocabulary and general reasoning or mathematical abilities to see that these populations are matched. So I'll tell you about neuroimaging data from people who are totally blind, born blind, and I'll compare those data to people who are sighted but are blindfolded during the fMRI scans, so that their current experience is matched but not their developmental history. And then I'll also say a little bit about people who become blind as adults and how plasticity in that population is quite different. So the first piece of data I want to tell you about is work by graduate student who graduated from my lab, Rita Loyotile. So she did this project in collaboration with Rodri Cusack, where she presented blind and sighted participants with audio movies. So I'm showing you here a scene from Harry Potter, but participants in this experiment actually heard excerpts, about 6-minute excerpts from audio movies. And what you, in this kind of study, the participants, instead of getting an experimental task and a controlled experimental task, they're hearing this rich stimulus that has language and plot and auditory motion and the sounds of objects, all sorts of things going on. And so what you can do with these kinds of stimuli is you can correlate brain activity across different people. What you can find is that if you correlate activity across different individuals, in sighted people, what has previously been shown is that higher cognitive networks in temporal and parietal and prefrontal cortices are synchronized by these conceptually rich, plot-rich semantic stimuli. By contrast, if you provide the same participants with stimuli that are degraded. So for example, you can, in the experiment that we did, we played a comedic routine backwards as a control stimulus. What you find is that instead, only low-level sensory regions are synchronized. So what we wanted to know was, in people who are blind, do the visual cortices behave like higher level cognitive areas? Do they need meaning to synchronize, or do they behave like low level sensory areas so that they will synchronize even for just perceptually rich stimuli? So these are the data that I'm going to first show you. So first, I'm showing you fMRI data correlating brain activity for a comedic routine played backwards to a group of sighted people. And what you can see, now remember, this is just an auditory stimulus. There's no visual information on the screen. And actually, the sighted participants are blindfolded. This kind of stimulus synchronizes the auditory cortices of sighted people. By contrast, if you give the same participants an audio movie, the auditory track from movies, it synchronizes lateral temporal, prefrontal, precuneus areas that are involved in semantics. So of course, the question is what happens in blind people? And what happens is that for the backward speech stimulus, you see very little synchrony, none in the whole brain analysis in the visual cortices. By contrast, most of the visual system is synchronized across blind individuals by these naturalistic auditory movies, which suggests that, they're responding to high-level semantic information potentially, but also they're engaged in these passive tasks that involve naturalistic stimuli. Lisa Muzz, who's a postdoc in my laboratory in collaboration with Janice Chen, took this work a step further and showed that you can actually, using multivoxel pattern analysis, if you divide these stimuli into short 10-second segments, you can differentiate the segments from each other based on patterns of activation in the visual cortices of blind people. So what that means is that not only are the visual cortices of different blind individuals co-fluctuating over time, but patterns of activity in the visual cortices contain information that distinguishes different segments of these audio movies from each other. So one thing about audio movies is that they're rich and wonderful and interesting, but of course, everything is confounded with everything because they're naturalistic stimuli. So we've also done more controlled experiments in the lab, and I'm going to try to whiz through this. So in one experiment, we showed that blind individuals activate visual cortices during sentence processing tasks, during auditory sentence processing tasks. So these are data from blind participants doing a sentence comprehension task compared to listening to lists of non-words and doing a memory task with these non-words. And what you find is increased activity in the visual cortices of blind people, but not sighted people for the sentences. Sighted people just activate classic language areas. And also, interestingly, in the blind participants, their responses in visual cortex to the grammatical complexity of the sentences. So not just that it's responding to sentences, but it cares about how grammatically complex the sentences are. And of course, you don't see this in people who are sighted. So we have not discovered a mysterious property of the visual system, which is that, it does not do syntax in sighted blindfolded people. Shipra Kangelia, who was also a graduate student in my laboratory, did beautiful work to show that these effects are not particular to language. So for any one higher cognitive function, you could come up with an ad hoc explanation. So you could say, the reason the visual cortex is involved in sentence processing in blind people is that sentences share some structural property with vision that we hadn't really thought of, right? Like maybe we just don't know what it is, but there's something there. But it turns out that this effect is not unique to sentences, and I will only give you one more example, but there are others. So what Shipra showed is that a different part of the visual cortex responds to mathematical equations. So when blind people solve math equations, the red portion of the visual cortex shown on the slide responds more to math equations than sentences. And activity in this math equation sensitive part of the visual cortex increases roughly linearly as the equation gets harder. So if you increase the numbers or you move the x to the wrong side, activity goes up in this part of the brain in blind but not sighted people. Again, these are congenitally blind people. So different parts of the visual cortex respond to math versus language in this population, which also shows specialization of this part of the brain. Together with Rashi Pant, Shipra showed that these kinds of nuanced cognitive responses in the visual system of adult onset blind people, of congenitally blind people, they follow a critical period of development. So what Shipra and Rashi did is recruit a group of people who are also totally blind and matched in current vision to the congenitally blind participants. But this group of people lost their vision at age 18 or later. And in this population, although you see a response to this auditory math task in the visual system, you don't see the interesting kinds of cognitive effects that we see in people who are born blind. You don't see, for example, sensitivity to the difficulty of the math equations. In a separate study, we also find that you don't see sensitivity to the grammatical complexity of sentences. So what it seems like is that the visual cortex of people who are born blind acquires higher cognitive functions, and what I would suggest is that it develops sensitivity to nuanced higher cognitive information, including difficulty of math equations and also grammatical properties of sentences. And I think that goes beyond the repurposing of domain-specific maps. I think it involves the obliteration of domain-specific maps and the replacement of these maps by other kinds of information. So that's blindness. And there are different ways of thinking about plasticity and blindness. I think of blindness as one kind of experiential adaptation, and but it's a particular example. Another kind of example that we could take that's quite different is the acquisition of cultural skills. So what I'm going to tell you about now is Yunfei Liu's work, who's a very talented graduate student currently working in my lab, who's interested in how the human brain enables coding, right? The ability to program. And coding is very different from congenital blindness in a number of ways. So first of all, coding involves not the loss of information from one modality, but rather the learning of a cultural skill. Although, of course, blindness involves learning cultural skills too, which I don't have time to tell you about, like Braille. But the part that we were looking at is the loss of visual input to the visual cortices. But this is a gain of a cultural skill. The other thing about coding, like many things that we learn it as adults, right? So We learn coding as adults. We learn calculus as adults. Many of us will take new physics classes or learn to learn lots of things when we're adults. And the question is, how does the brain enable this? Do we recycle domain-specific cortical maps for coding? So if we were to do so, if we were to recycle domain-specific cortical maps, I think the best candidate for that kind of recycling would be language, right? And various people have suggested this possibility. You know, many programming languages, Python being an example, use linguistic symbols to are used in, words and letters and symbols are used in Python. Python and programming languages also have various properties of language, including not only being symbolic, but also involving recursion and nesting and being hierarchical. So you could imagine that when you learn to code, you take language mechanisms and you recycle them. And what I'm going to tell you is that that's not what we find, right? And actually, we and others have found the same thing, which is that's not how coding works. So in this study, what Yunfei did is he recruited what I'm going to call Python experts. So people with expertise in Python coding. On average, these people have about 6 years of Python experience and about 10 years of coding experience in general. And so what he did in these, right, so we're going to ask what cortical mechanisms does coding recruit, and to what degree does it depend on language systems? And if not, what other neural systems are involved? And let me tell you in detail about that in a second. So in this experiment, he presented participants with snippets of code. So first there was a code comprehension phase where they got a function. And then there was an input phase where they got an input into that function. And then they were asked to say whether the output of the function was the thing that they saw, true or false, right? So that's the task. And the fMRI analyses are going to focus on the presentation phase. So the time that the participant is comprehending the function, but doesn't yet know the input. And what Yunfei did is he compared the coding condition to a memory control condition where participants see the same symbols, but now what they have to do is not understand the function, but rather to just remember all the symbols. That we're going to be asked if the same line of symbols occurred previously, okay? So when he compared this real code to fake code, what he found was a frontoparietal network, interestingly, a left lateralized frontoparietal network that responded more to real code than fake code in these participants. What he further showed is that this network doesn't just respond to real more than fake code, actually it encodes information that is relevant to code. So what he tucked into his coding trials is two different kinds of trials. So if functions and for loops. And what he was able to show is that based on patterns of activity in the prefrontal cortex, as well as the intraparietal sulcus, and a portion of the middle temporal gyrus, he could differentiate for from if functions, suggesting that they're really involved in representing code relevant information. So what Yunfei also did is have the same participants do a bunch of what we call localizer tasks, right? So these participants also did a language task, a math task, and a logic task in a separate scan. So let me just show you. Let's see if I have the kind of stimuli that they saw. So for the language task, they saw sentences like the cat chased the girl that likes the boy, and another sentence that they were asked whether in passive sentence, if they mean the same thing. For math, they got equations and they had to say whether the value of X was the same across the equations. And then for the logic task, they got statements like if either not K or not T, then B, if B, then either not K or not T. And they had to ask if the first statement implied the first. The first statement implied the second. So using this localizer, he was able to identify parts of the brain that are preferentially responsive to language, parts of the brain that are preferentially responsive to math, and those parts of the brain that are preferentially responsive to logic. And what he found was that code comprehension overlapped most clearly with logic. And the overlap with language was quite low. The highest overlap shown in the green bar here and also circled on the left is the overlap between the logic and coding, shown in blue. The very low overlap is observed with language. And then there's some intermediate overlap in the left hemisphere, at least, with the math functions. So what do I conclude from these data? So first of all, I guess one thing that I should say is that these frontal parietal areas that we observe in the current study that are involved in logical reasoning are classic working memory areas. And actually a study by Ivanova in Evelina Fedorenko's lab that was published concurrently with our experiment showed that this coding overlaps with central executive mechanisms in frontoparietal cortices, which are also engaged in logical reasoning. So I think what all of this work suggests is that these domain general or general-ish, I don't think there's any such thing as fully domain general, reasoning mechanisms are recycled for code comprehension, and they are flexible enough to represent varied kinds of information, including not only logical reasoning, but also code. So here are some overall conclusions. So the first thing I'll say from the blindness data is that early in life, domain-specific maps can be entirely overwritten. So the human cortex at birth, I think, is highly flexible and pluripotent and can take on a range of cognitive operations and assume a range of cognitive maps. There are limitations on plasticity in domain-specific cortical maps in adulthood. So as far as I know, there actually isn't any evidence in adult brains of really dramatic reorganization of cortical maps in adults. And another way in which humans are extremely flexible is not just by adapting domain-specific cortical maps and domain-specific mechanisms like language or vision or numerical processing, but actually by using a powerful domain general reasoning system that allows us to do all sorts of things like coding, and probably also plays an important role, not only logical reasoning, but also in mathematics. So with that, I want to finish and just thank all of the wonderful people who did this work. I want to highlight Yunifei Liu, who did the coding work, Shipra, who did the work on number, Connor on language, Lisa and Rita, and the entire lovely lab that works on all these projects. And of course, thank the blind community for their contribution to this research. We couldn't do it without them, and our funding sources. And thank you guys for listening to the talk.
Thank you, Marina.
My pleasure.
We can't hear you, Ida.
Oh, I'm asking if you remember how to oh, perfect.
Now we hear you.
Exactly.
Yeah.
Yeah, perfect. Thank you so much. It was so fantastic to see your results. We have already some questions, but usually John and I first start asking questions. And since you're John's one of John's favorite people, I allow John to ask his questions first. And I want to remind everyone to, oh, and you can see there is applause going on in the chat. I want to remind everyone to add your questions to ask a question area. Also add, maybe ask for me if you don't want to show up. Otherwise, if you have a high voted question, I will ask you whether you'd like to join the screen, especially if it's related to a conversation that we're having. and we can start to go to audience questions in about like 20 minutes after John and I are kind of exhaust the conversation on this end and move on to this. John, take it away.
So, Marina, this is an area that, as I said in the introduction, I care a lot about. And I actually, full disclosure, I mean, you and I have talked in email, Tamar Macon and I are writing a very large article that never seems to end on questioning this notion of cortical plasticity, right? And it's not that the story isn't fascinating, right? That something is happening that is different early and late between adults and the congenitally blind. What we question, and I want to really push you on this, is that when you say obliteration, total repurposing, I just question whether in fact the data support that idea, right? In other words, I think originally, to tell me if I'm wrong, people thought, well, if you're blind, then, V1 should be a place that is no longer useful, you're blind, and therefore V1 should be the place that should be ripe for repurposing. But that's not turned out to be true, right? It's the higher order visual areas that seem to be repurposed. And there are many results, right? For example, if you teach Braille to a sighted person, they will activate the visual word form area, right? So, I'm not going to go through all the data in other areas, but what seems to be the case is that it's areas that are already polymodal that get upregulated, not primary visual cortex, right? And that what you seem to be doing, and the position, and I'm open, this is for the fun of debate, as Ida said, is that what you're seeing is an upregulation of architecture that already has that capacity, not qualitative switching of identity of cortical areas. And I think that's compatible with the fact that if you really took over all these areas, that were the famous experiments on cataracts, they would not be able to see when their cataracts are removed up until the age of 12, and they can. So I think the story is much more likely to be some age-related gain modulation of already latently polymodal cortex in higher order areas than true rubbing out of identity of a region by others, which is why whether it's A1, M1, V1, on careful re-examination, there was already evidence for these responses in the healthy brain. Okay, so my position, and I'd like to press you on the results you showed, is that nothing shows a qualitative switch of identity rather than potentially an increase in the gain on latent pre-existing circuitry. So that's sort of what I'd like to get your view on. Now, on the, well, maybe before we get to the coding bit, because that doesn't seem fair for me to send this shot across the bow and then switch to the coding, which is a different issue. But
maybe we get to hear what Marina says, because
this
is very
I'd rather, so in other words, can you really prove genuine qualitative switching of identity?
Okay, so that's a great question, and I think let's talk about it. And I look forward to reading your paper with Tamar. And I will say that, there are so many cases of adaptation in humans, and Tamar and you have been working on adaptation in the motor system, and we can talk about how different examples, different things happen in different cases, and they tell us that there isn't a single way that the human brain adapts. So that's another interesting question to discuss. But let's talk about the blindness case for a second. And you said a couple of particular things that I think are just worth addressing. So the first thing you said is something about V1 being not adapting. So in the case of blindness, V1 is actually thought to be one of the more drastically changing places rather than secondary visual areas. So here I'm just showing you, I don't know if you can see my screen. These are data from the study by Lane, which I was telling you about during the talk, showing responses to grammatically complex in Black sentences, less grammatically complex sentences in gray, and non-words in white in V1, and the lingual gyrus. And what you can see is that, yeah, we see these effects in V1. And in fact, some people have suggested that because V1 is a really early visual area, sorry, I'm just, closing this file so it's not on the screen. Actually, it adapts more because it's not getting any domain specific or a relevant input.
Sorry, we're seeing your grant notes, by the way.
Sorry,
okay.
They're rather boring.
But just to be clear,
you're welcome to them. So, but the first, so that's the first thing is about. So let me just go through the list. So the first thing is V1. So it adapts. I know there's some discussion in the deafness literature where the case may be quite different, but V1. Many of the effects, and not just our lab, lots of labs have shown that V1 is repurposing for all sorts of things. And people like Amir Mehdi have even suggested that maybe it's only V1 that repurpose. I don't see that kind of stuff in the data, but there we are. The case of the VWFA is actually a very interesting one. And we have a line of work on Braille going on right now to try to understand it. And I would say that the jury's most definitely still out on how Braille reading works. And actually, we find some evidence that, in fact, parietal mechanisms are developing what I would term tactile word form areas, or I don't know if they're areas, I don't know if they're word form, but are developing specialized Braille reading regions in blind people that don't exist in people who are sighted. Exactly what the VWFA does in people who are blind is unclear. It might do, like we find, for example, that if you localize the VWFA using Braille reading, you find syntax responses in the VWFA of blind but not sighted people. So I think in the case of blindness in particular, which is the one, that I'm, that one, I think there's pretty clear evidence of reorganization. Now, there are other things that we can talk about, like what would it take? What kind of evidence would it actually take to convince us that something was being obliterated? What are we missing? And there's lots of things missing, actually. So, for example, I think there's some evidence for behavioral relevance from TMS. I think it could be much better, right? So, do we have evidence for actual maps? I think the evidence is like, okay, we could have better evidence for actual maps in the visual cortices of blind people, but the evidence that even the earliest visual areas are responding to information they have no business responding to is quite solid, I think.
Again, sorry, just on that. Oh, sorry, Ida, go ahead.
I just want to push back on this a little and maybe ask for a little more precision. You use the word the visual cortex does syntax a couple of times in your talk. That's a very strong claim. So that would require at least to show that temporally, if you had a syntax error in front of a person, that after about, I don't know, what is the lowest number of milliseconds? I don't know if it's 300 milliseconds. This is like P or N 300, whatever it is that it takes for the error response, that response should register in their visual cortex. I'm not sure exactly if you have looked at whether some other regions are doing this and maybe some parts of the visual cortex are reflecting that. Do you have examples that perhaps somebody got damage to their visual cortex and was congenially blind? Did they lose their language? Did they have cognitive deficits? Did they have inability to do math? Like, did these things actually happen, because otherwise, I mean, I have seen the visual cortex show up in many situations where they didn't have any business, when the situations are not properly controlled, even when the subjects are closing their eyes or such. So there is a kind of a temporal dynamic to it anyway. So for instance, if you show a sentence to me that's grammatically correct and one that's nonsense, I might look at one more, and I might process it more, and it might engage my attention more, and therefore the internal dynamics of my brain will synchronize in a different way, and you can see it in many parts of my brain. I worry that some of the effects that you showed us, I didn't see whether there was a control. I guess maybe there wasn't enough time, but I didn't see the meticulous control in the way that we would have to do in fMRI to make strong claims. So I got really worried when things went that way. And even when you showed us that syntax study, it seemed like the brain is very masked because we were only seeing a small part of the visual cortex. Whether it was or not, that's not the issue. The issue is I don't know how long they were looking at this. I don't know if they had more time to look at one versus the other. I don't know if they had more time to process it. I don't know what was happening in other parts of the brain, because it's not possible that only the visual cortex was responding to syntax and not anything else, right? So I'm a little concerned about what is going on with the rest of the brain. What is the connectivity of these visual regions with the rest of the brain? And does it follow them or does it start? Because if it's following them, it could be used as an auxiliary part. But if it's leading, that's a whole other kind of story. If all of a sudden I'm using and maybe it is, I'm not saying it's not entirely, I'm not saying it's entirely impossible. I'm totally open to somebody showing me anything is possible in the world. I just need the evidence, so to speak. So it's not that I'm fundamentally or theoretically against it. I just feel like the evidence was very scarce for that. That's my perception.
And Marina, just to add, because I'm also horrified. I completely agree with Ida on that. So let me give examples as why I just don't think that activations are really telling us.
I think a lot of things are getting said, and I'm happy to
go ahead.
Totally fun, but I think
totally fair.
Yeah, we should get information right. So, so I'm going to start with the smaller thing. So you said a lot of stuff, right? You said something about the particular study and what was being masked and what was being shown, and then also you said something about time and connectivity and functional relevance. So
it wasn't too many things. Just to summarize it, very simple things, right? It's not too many things.
Okay, I'll keep going. So I'm going to go through each of them because I think they're kind of different. So the first thing I want to say is, which I feel kind of is important to me, is that, so the study that I showed, there's absolutely nothing masked in that study. It's just showing whole brain activation. I'm sorry that I went so fast, but the participants in that study are listening to sentences. So they're not looking at anything. Neither the sighted people or the blind people are looking at anything. They're hearing the sentences. They hear a sentence, they hear a grammatically complex one, a grammatically simpler one that is exactly the same sentence counterbalanced across people, but with a movement dependency. And then they hear a list of non-words. Those are the conditions. So there's no looking, there's nothing like that. And so what you find in blind participants, but not in sighted people, is and we've now done it in larger samples and in other groups of people, larger responses to sentences than non-words in the blind participants, but importantly also responses to the movement dependency in the blind but not the sighted participants. It has nothing to do with looking. It has nothing to do with the abstractness of the sentences, because the sentences are matched in their content, and everything to do with their grammatical content. So I think those data don't completely answer all the questions that you asked. They just are what they are, right?
I'm sorry. So because it was just introduced, and I think it wouldn't be constructive to go over all of it, but still, there are so many questions there, whether there's a confound or not, I still don't know. I'm pretty sure it wasn't just the visual cortex that showed up in response to something.
Oh, absolutely, yes.
Yeah,
and this is important.
But sorry, I want to make sure that we get to the more broad thing, and because otherwise we can, ad infinitum, bring up a new study and spend many times just understanding that, because unfortunately I haven't read that, and at this point I think it might be like We can definitely discuss it, but I want to go back to the big picture thing that I'm saying.
Yeah.
Do we at all have enough evidence, assuming even there was no confound in these fMRI studies, and they were all completely controlled for everything? Do we really have enough evidence to say the visual cortex is doing syntax? And I'm going to repeat what I said about the temporal dynamics. Does the reaction to a particular error, and this is not something you can figure out in fMRI, this is something that's much faster, it's at the level of hundreds of milliseconds, and it needs to be done with a method that is sensitive to that. Does the visual cortex show those responses before the language areas or frontoparietal areas, or after, or at the same time? Because I think even little milliseconds of, sorry, did I interrupt you?
Okay, I don't mind.
No, but I wonder if I did, because like I'm not sure, I'm confused a little bit. I'm trying to make sure that the discussion this was not an interruption in the sense I'm trying to.
I don't mind. It's fine.
Okay, great. Yeah, somebody mentioned something in the chat. I want to make sure that the discussion goes towards the bigger picture question, and we don't.
I'm happy to. Yeah, I get what you're saying about timing, so I'm happy to answer that question.
Yeah.
So I guess I would say, you know, so the first thing is I don't want to like. It's important not to overstate the data that we have. So, you know, there is really not that much work in this population, and we have limited information, right? So the new work is constantly being done by our lab and other labs, but there's some limitations, right? So with regard to timing, there isn't a lot known about timing. And I should say, it's very important to say that it's not the case that in blind people, the classic language areas are replaced. They're not, right? Because the blind participants show also Broca's area responses, and we've looked and they look typical. They also show temporal lobe language area responses. Actually, blind individuals show much more variable lateralization patterns, but apart from that, the frontotemporal networks look similar in blind and sighted people. So what I think is actually happening is the language network is expanding rather than like replacing something. We don't have clear timing information. It's not actually as easy to do as one might think. To even in classic language areas like in frontal and temporal cortices, the temporally resolved measures that we have are actually not great at spatial localization, especially in cases where a really distributed large network is involved, like in the case of language. So it's not totally trivial to figure it out. There's a little bit of work by Bridget Roeder showing N400 responses that are like pushed towards the visual cortex in people who are blind. I think one of the most exciting pieces of work is by Amir's group showing that you can impair verb generation in people who are blind by stimulating the visual cortex. So that study was done like many years now ago, and the kinds of errors that blind people make during that kind of stimulation is semantic errors. So that suggests that there's some functional relevance of this part of the brain to language processing. But you know, I totally agree with you. There's certainly limitations on what we know. I guess I would say that the degree, you know, what kind of evidence. We have multiple hypotheses, multiple possible worlds, right? And I think we probably have different priors on which hypotheses are most probable, and then we can evaluate the evidence and like change our probabilities on the hypotheses. So one prior would be like each brain area is fixed to do a particular cognitive function. You've got to just show me absolutely everything, you know, to the kind of weight of evidence I would need to show that a brain region has changed its function across domains would be, you know, I need more, whatever it is that I need. So that's fine. Another possibility is that, you know, in each individual, I guess my prior is the brain adapts itself across people to the cognitive niche that the person has. So if the person has no use for certain functions, then the part of the brain adapts to do whatever functions their brain does have use for. And then this population one of the functions that it has use for is linguistic communication. But I totally agree with you. There's much more TMS work needed. There's useful work to be done using measures like MEG. So but I think the evidence for this population in my mind is much more in favor of the idea that that the visual cortex like V1 is doing higher cognitive things. With regard to connectivity, which is something you asked about, in people who are blind, visual areas, including V1, but also secondary visual areas, become synchronized with frontoparietal networks, much more so than in sighted people, and decrease their correlations with auditory and sensory motor cortices. So that kind of goes along with the idea that what's happening is that these top-down networks are incorporating them. But again, like, yeah, that's what we know. That's what I've got so far.
Sorry, I'm going to ask you again then. Oh no, John, do you want to go ahead? But I'm going to.
Yeah, I just wanted to. I think this is an extremely important area. I mean, again, I do not agree to the degree that qualitative change in the input-output tuning of an area where it takes on a qualitatively different computational function has been definitively shown. I just don't think that's true. I think that when you look at the data that we've painstakingly gone through, it looks much more like upregulation of latent architecture that always had that computational capacity, right? And I don't think that imaging experiments or TMS perturbations can break the tie there. I'll give an example. If you take in the motor domain, if you take a mouse or a monkey and you lesion motor cortex, you can upregulate premotor cortex. And then if you lesion that premotor cortex, you get a recapitulation of the deficit. But you never get if you lesion that premotor cortex when M1 is intact. But that doesn't mean that that premotor cortex suddenly took on a new identity. It's much more consistent with what has actually been shown is you simply facilitate and upregulate that latent circuitry when it's had other areas have been damaged. So I just don't see how either imaging, you can decode from many parts of the brain limb movements, but if you lesion those parts of the brain, limb movements are affected at all, right? So I think there's a big difference between showing activations at lower thresholds, showing the ability to decode from unexpected areas, but those kind of evidence do not prove that that area has qualitatively switched identities. And I think reorganization implies that. It's some magical switch of the tuning functions of neurons or areas. And I just don't think, and I'd be open, that the data have shown that. And I did ask you about the cataract surgery. They're not suddenly unable to use their visual cortex because it was taken over by cognitive functions, right?
Yeah, I mean, the cataract case is really interesting. I mean, the case that I tend to think about is Michael May, who's a really interesting, I don't know if Ione Fine's work with this individual who, due to an accident, lost his vision at about age 2.5, and then had a corneal transplant to restore function in one eye. And what Ione has documented over the years is that this individual has profound visual perception deficits. So it's not that he doesn't see, he does see something, but he cannot recognize faces, he cannot tell facial expressions, he has trouble with object recognition, like personal reports that I've read say things, he's a skier and has been a blind skier all his life. And the personal reports I read was that this was a while ago, so I don't know if he's still doing this, but he was skiing with his eyes closed because he was doing it fine before. So on the one hand, I agree with you. I don't think these areas have lost the capacity to do some visual function. They still have connectivity to those to visual input in these individuals. But I think there's also quite a bit of evidence that they've lost a lot of their capacity to do vision well. Right. So I don't know if that, I do
But there are
think
two ways to interpret that, right, Marina? One is that they never really got visual cortex back to doing what visual cortex does, versus because it was already crowded out by cognitive functions, right? In other words, I mean, and that leads me just to my final question, because I think, I mean, which I really wanted to ask you a question. Let's say that one of the, Barbara Landau, who you and I know very well, right, and the work that she's been doing with Lisa Newport in Washington, right, at Georgetown. What Barbara told me is it's really interesting that if you look at congenital stroke, where they basically lose a hemisphere, and then you see the homologous region activating with language acquisition, what's really interesting is it's the exact homologue that activates, not just cortex in general being pluripotent. So in other words, there are two ways to interpret those results. One is that there's no plasticity whatsoever. You have to use the exact homologue in order to actually get language function. It's not like you can, which means that it's the reverse of plasticity. It's got to have the same connectivity, the same input-output relationships in order to be able to do the takeover.
Yeah,
right?
I would say, I think that's a really interesting case, John. And I don't think there's really any way to argue that in that case, there's no plasticity whatsoever. At least that's my take on it. So here's what I would say. If you take an adult, if you take 97% of adults and they suffer from a stroke to their left hemisphere, a massive stroke, they will be aphasic for the
So
rest
that was
of
my
their life.
question. That was my question
Right.
to you. Yeah.
Right. So okay. But if you take a child, an infant, and they have their left hemisphere removed in 90 plus, and Lisa Newport will tell you this, they develop normal language function.
Right.
As far as you can tell, right?
Yeah.
So
but you're missing my point. But that's not real.
No, I know. I'm not missing your point. I'm disagreeing with it. I'm not missing it. So what I would say is, but you're totally correct. What they're using to do language from the imaging studies that we have is the right homologue of those areas. So there's multiple ways of looking at that, right?
Yeah.
One is that these areas have the potential to do language, but don't do it in the adult, or don't do it in most people, but do it in this subpopulation. So I don't think that's evidence for no plasticity, but of course there's still a question about what makes them land in those particular areas, regardless of hemisphere.
Right. So but just on that point, so you'll understand, you're absolutely right that it's a fascinating mystery why it is that if that homologue exists with its latent capacity in the adult, why can't it be upregulated the way it is in the children, right? That is absolutely true. And I don't think we know the answer to that. But there's a difference between the absence of the ability to upregulate latent homologue regions versus reorganization in the strong sense, where a region that never had the latent capacity suddenly becomes, through some Ovid-like metamorphosis, a new region altogether. And I think that the fascination with respect to plasticity remains, whether it's the view I'm taking, which is an amazing ability that wanes with maturity to upregulate latent shared homologous computational structure, versus what I think that you were saying, at least in the case of V1, of a total change of identity, right?
So I don't think in either of these cases it's upregulation. That's my thought. I don't think that's what's happening. I mean, and I don't think the evidence can, in the case of the language reorganization in cases of pediatric stroke, the current evidence can't distinguish between your argument and mine. But my argument is that no, it's not upregulation, and you don't have a right hemisphere area just sitting there all your life as an adult, like capable of doing language, but not doing it, right? So what I think is that it's not doing it, and it's not capable of doing it because it never learned to do it when you were a baby. But this is an important point, which is that I don't think what I'm arguing, even in the blindness case, is for some kind of like equipotentiality, anything can do anything. That's not at all the argument. And I think there's clear systematicity. So in fact, what I think is happening in the blindness case is that people have been thinking about what happens in cases of plasticity and blindness by thinking about what the individual cortical area does, and then trying to, in the sighted person, and then trying to guess what it does in the blind person by doing that. And I think that's fundamentally misguided, right? So what I think actually is we should think about is the connectivity to this part of the brain. And actually, most of the connectivity to this part of the brain in a person, in sighted and blind people, comes either from other visual areas, and of course from the LGN, but also from frontal, parietal, temporal areas, not from A1, not from primary motor cortex, not from other primary sensory systems. So what I think happens is when you remove input from the LGN, it's the parts of the brain that have the strongest connectivity that end up taking over. So that's one, it's not equipotential. The second really important point is that I think like recalling Mar. So I don't think that in order to get fundamental changes in cognitive function and content, you must have fundamental changes in structure. So I think the same structure can represent a lot of very varied content. So I don't think there's anything magical about what's going on here. It's just that a neuron is a neuron is a neuron in the human brain. So that's the argument here, right? The argument is, and that's how it connects to coding and other kinds of cultural adaptations. Why is it that we can thrive in New York City when what we evolved in looks nothing like that? Because our brain is capable of updating what it does. That's part of the reason. So that's what I would, that's my argument.
No, I'm going to just say one last thing.
John, do you mind if I very quickly touch on that? So Marina, you do this rhetoric move where at the end of every time that you end something, you say a kind of a less controversial statement. Yes, I think we agree that human brains are adaptive, but let's go to the actual question. So in terms of connectivity, I totally agree with you. I have personally seen the connectivity between visual cortex and frontal parietal regions, but it still remains the question. So I think it's very reasonable what you're saying, which is that the reason for these areas to appear to be recruited or be more active in certain functions that are associated with frontal parietal region is because they are thusly situated within the connectivity of the brain. I think that's a great reason for it. There is also a hierarchy to the visual cortex. Is that kind of hierarchy also preserved either in terms of the time scales or in terms of the abstraction? So that's one question that I have. And also, again, in terms of the connectivity, if it is the connectivity that it's driving it, do they become a necessary part of that processing or not? So if I am a blind adult and I lose my visual cortex, do I get impaired in language or not?
Yeah, so you asked about, sorry, one is the necessary, and sorry, what was that? There was a previous,
okay, I'll ask one by one.
No, sorry, I had it.
No, I'll ask one by one.
No, yes, it was upregulation, maybe? No, anyway.
No, I didn't, I don't use the word upregulation. So I'm gonna use.
Oh, sorry, can you just restate?
Yes,
or
happy to say it again, totally fine. So my first question was, is the hierarchy in the visual cortex preserved?
Oh, great.
And the hierarchy has different connectivity to different parts of the brain. Do they appear in this connectivity at the same time as the PFC or after it? So those are the two things that are about the hierarchy and connectivity combination.
Got it. Okay, so the first thing about you asked about the brain damage case, and unfortunately we don't have that kind of evidence. I would love to get that kind of evidence, but it requires, I hope that no congenitally blind person ever has a stroke that affects their visual system. But the reality is, like, we would need to find this 0.001% of the population intersecting with another 0.0001. So I hope that by the end of my career, if that person already exists, they will end up either in my lab or someone else's. But the closest thing we have right now is using TMS. So yeah, and we're going to do that. We're going to try. And so there's been already some work, and we're trying to follow up on it, right? With regard to the hierarchy, no. So there's no evidence that the like there's a cognitive hierarchy of processing of the kind that exists in the visual system inside of people and people who are blind. Actually, there's been some people who, I think maybe Uri Hassan suggested, I can't remember actually who it was, who've like suggested maybe there's a flipping of the hierarchy where now like the visual cortex is the top of the hierarchy, whereas it was the bottom and the sighted people. We don't actually see evidence for that either. There are interesting differences between the kind of reorganization that you see in early visual areas and secondary visual areas. And I think secondary visual areas, which have domain-specific functions and people who are sighted, like the VWFA and the PPA, for example, are preserve more elements of their function than non-domain specific areas, although I don't think they preserve the functions altogether, but they show some more similarity between what they do in a sighted person and what they do in a blind person. I don't know if that actually was, I'm not sure if that was your question, but yeah, that's what if it is, then that's the answer.
And before Lawrence comes in, just to the point that, Ida made, where, no one denies adaptability, right? In other words, reorganization is a word in the literature that is reserved mainly for lesions and disease, blindness, amputees, stroke. The word reorganization is very rarely used to study, I don't know, violin playing in healthy people or reading. So reorganization has this extra implication of identity change in regions of the brain where they're doing things they didn't do before. It's very rare, I haven't seen it, the word reorganization to be used for the adaptable plasticity where we didn't evolve to play the violin or play tennis or to read, but we can learn to do that with presumably recycled computational structures. So I think reorganization has a qualitative feel to it that is different from adaptability, plasticity, and recycling. I don't feel like those should be used interchangeably. And the only discussion being made here is there something special about injury, especially congenitally, that leads to a special superpowered form of plasticity that cannot be, which is worthy of the word reorganization, that isn't used in adults, whether they've had injury or whether they're just learning new skills. And it's the implication that there's something special and qualitatively different that I think we're questioning, rather than there's an ability to adapt, recycle, learn new skills. Okay, because of course you're right about that. It's whether there's something special that deserves the term reorganization that we're wondering about.
I think maybe Lawrence's question and then David's question are directly related to this conversation. So Lawrence, do you want to go first, given you have timeline?
Yeah, definitely. Can you hear me okay? Yeah, great. Yeah, so I mean, I guess it kind of links back to what Ida was asking about the temple hierarchy, and you may have touched upon it already I heard you mentioning Uri Hassan's work, but then it cut out as I was joining. But my understanding of some of that work with Hassan and Baldessano is that if you go and look in visual cortex during movie watching, the activity tends to be very kind of changeable across time as they're watching the movies. And then as they progress up to higher cortical areas, it becomes more temporally sustained over time. And that's always kind of been linked in my mind, not only to the connectivity to visual input, as you're arguing, but also to things to do with the intrinsic properties of those cortical areas. So there's all of that nice work by Xiaojing Wang on intrinsic time constants of autocorrelation across the cortical hierarchy, arguing that some of the microcircuit properties of these primary visual areas mean that they are adapted to process temporal information over kind of shorter time windows. And so I guess my questions for you are, in the auditory listening case, in the congenitally blind individuals, do you see the sustained activity across time now back in visual cortex? Do you see kind of like long temporal windows of integration in that primary visual cortex? And do you also see that if you just look at like resting data in those individuals as well?
Yeah, that's a great question. And the naturalistic auditory movie experiment kind of gets directly or the closest to what you're asking. And so based on those data, what I would suggest is that, in fact, this temporal integration window is a functional property, not an anatomical property of visual cortex, because in the blind participants, it's sensitive to, you know, long plot relevant elements rather than the short windows, whereas in the sight of people, at least for visual stimuli, it's the flip. So I think in answer to your question, it seems to be a functional rather than an anatomical property. But that's like one kind of measurement, right? And of course, all the kinds of things that we're looking at are much slower than what the visual cortex usually does, including not only these movies and the sensitivity to long time scales, but also, you know, sentences are much longer than, you know, yeah. But I guess one thing I would say is that we don't have any evidence that there aren't influences of the anatomy. I just think they're probably more subtle than we would have expected. So I think the microcircuitry of the anatomy could, for example, determine how well that part of the brain does something, which you don't really have an opportunity to test because, as I said, you know, these participants, it's not just that they rely only on their visual cortex to do these functions, they also have all the typical areas. So we don't know, and we couldn't ever really know what would happen if they only had their visual cortex. That same TMS study that I was telling you about by Amir showed that blind people were more resilient to stimulation of the frontal lobe during making verb generation errors. Again, it's one study, but that kind of suggests that, you know, that visual cortex again is contributing something, and maybe, you know, able to supplement the function of other areas. But I don't think we know. It could be that there are these subtle these differences that have effects on how well something can do a function, a cortical area can do a function, but don't categorically determine what that function is going to be. There's like this mismatch between how well it can do something and then what the function ends up being. The other thing to mention is like developmentally, the visual cortex of people who are blind may not have ever developed language functionality had these people not been born with Broca's and temporal areas that evolved for language. So that's like another kind of thing to point out. So there's all these complexities. Nevertheless, I think that what the data do show is that the kind of thing you're talking about is a functional property, and the cognitive content of what the area is responding to is at least very early in life experience dependent.
And do we also know anything about whether that's reflected in changes just in resting activity if they're not doing any kind of task as well?
Yeah, so I am not aware. So there's lots of resting state studies in people who are blind, but I'm not aware of anything that's looking at like the time structure of correlations. If you have ideas for how to do that, I would love to hear about them. That would be great. The only things that I know now are looking at synchrony. And for what it's worth, you know, the visual cortices of people born blind is synchronized with higher cognitive networks. Does that tell us that they are, you know, working at a slower time constant? I don't think so. So yeah, I don't know of any work that shows what you're saying. Yeah.
Towards Lawrence's question, and I'm very grateful for the questions, Lawrence. What is the longest time scale you observed in V1 in the congenially blind?
Sorry, you're asking me?
Yes.
Okay.
Yes. I don't think Lawrence has worked on congenially blind, but
so for example, you get
okay.
Yeah, sorry, you get higher synchrony for intact narratives than chopped up sentences, and higher synchrony for chopped up sentences chopped out of a narrative than for like backward speech, so that has no long range temporal structure. So that's the kind of measurements that these are.
Sorry, I was referring to the explicit timescale measurements of the type of stuff that Janice, the Hassan Lab, etc. have done, where you can see, you can measure basically what is the longest while watching a movie or parsing a sentence, a particular region might be showing.
That's how these measurements are made.
Right.
So the way these measurements are taken is by chopping up the stimulus and shuffling it progressively. So although it's phrased in terms of like how long is the time constant? The answer in terms of the data is that, you know, if it synchronizes more for narratives than chopped up sentences, that it's sensitive to the slow fluctuation of content that is beyond the length of a sentence. But again, since I think that's a functional property, I tend to just talk about it in terms of the content of the stimulus rather than the time. But that's the answer to your question, is like the slowest ones that they've really talked about are the ones that it's sensitive to. That's the answer to your question.
David.
So, sorry, one second.
Okay.
Right. So I've worked with that data. I've taught summer schools with that data and chopping it off. What is the longest chopping off? You're saying that the longest possible that they ever used is the longest that you found in V1. That's what you're saying?
Yes.
Okay. And in the
so
human...
we've only done one study with blind participants with these kind of stimuli. So we haven't done all the cool things that you could possibly do. And
no, I'm
yeah,
just asking you about something specific. So let me just, sorry. So in V1, in the blind, you had it for until the largest time scale. And in humans, you had it until only the smallest one for V1.
Sorry, I got lost a little bit.
Yeah, so in those studies, they show that V1 had a short time scale, right? So yes. Without saying seconds or how long, which is what I was wondering about. So you're saying that in healthy adults or sighted adults, it was the shortest, and in the blind adults, it was the longest in V1, not the whole visual.
So in the study that we did, we're using auditory stimuli since we're matching the sighted and blind participants, and the sighted people are blindfolded. So the measurements of the time constant in sighted people come from other people's data in sighted people watching movies, right? So what we're doing is having blind and sighted participants listen to movies, and also listen to Pyman and listen to Pyman chopped up into sentences, and then Pyman played backwards, which is basically the original experiments. That's how they were done. And what you find is that synchrony is higher in visual cortex for people who are blind for the intact narratives, which is the slowest stimulus, than for any, for the chopped up to sentences Pyman, and then for backwards. In sighted people, there is no synchrony. It's minimal for auditory stimuli, irregardless of whether or not they're degraded or chopped up.
Right. They did have audio studies in the sighted adults, so you didn't compare it to the. Yeah, I think I need to take a look at the study. So I'm going to go to David Linden before we move too far from the animal versus human comparison. And so, and I just want to also maybe preface this by, at least in humans, the prefrontal cortex, just the cortical layers are different from visual cortex. And so there is connectivity, of course. So obviously, you're not suggesting that the cortex is also reorganized, right? That's not the level at which you're suggesting there is, or is it? Is it like a columnar reorganization or just functional?
It must be a functional one, because those the layering that you're referring to in the cell types are present at birth, right? And these are people who are born in the same, sighted and blind babies are born in the same way. So I think the way I take these data is more of a like dissociation between function and anatomy, than evidence for like anatomical, drastic anatomical change. Although probably there's evidence of some anatomical changes, but I think,
Right. So so for instance, I focus on prefrontal cortex and the hippocampus. We don't think everywhere can do the same things. So I'm just wondering, just to make sure, so given that you also agree that there is difference in layers in prefrontal regions and even subregions and the visual cortex, not everything that happens in the prefrontal cortex can be done by the visual cortex, right? Or do you think, no, everything that can be done by the visual cortex by the prefrontal cortex can be done by the visual cortex?
Yeah, so this relates to the thing I was talking about with Lawrence just a second ago, which is that I think, first of all, the hippocampus is a very different structure from the cortex. And so I wouldn't extrapolate from what we find to.
I did not. That was just a mention. No, let's put that aside. Not hippocampus.
Okay. So, but I do think with regard to cortex, V1 and prefrontal cortex is probably as different anatomically as you can get. So I don't think this is particular to language or to math or to nonverbal executive control, which is another domain that we've studied. I think it's common to all these domains. So I don't think it's anything special about which function is being transplanted. So what I think is that during development, the visual cortices can acquire functional responses to domains that are more like those of the frontal lobe than those typically observed in the visual cortices. What I think the evidence is weaker for is although functional relevance, right? And to what degree? So what I can't tell you is how well does the visual cortex do these functions? So I think it's assuming these functions in the sense that the neurons there are now responding to this information in a way that they don't in people who are sighted in a pretty fine-grained way. But the degree to which they can support these functions in and of themselves and how well they're doing it may depend on their cytoarchitecture. And I don't know I think we have limited information with regard to that question. I don't know, does that answer your question?
It doesn't, but I think I want to go to David Linden just to make sure, because he's been very patient. So David, please take it away.
Okay. So first of all, Marina, I thought that was lovely presentation, really thought-provoking, really interesting. It's, in these situations, we always start to dig in right away and focus on what our problems are. Let's step back and say, that was great. I really enjoyed it.
Thank you. Yeah, thank you very
much. The question I had was sort of sparked by your introduction, where you were correctly pointing out the human-specific, human-enhanced functions like math and language. And then you used that as your introduction to cortical plasticity and the effects you see comparing sighted and blind people. And I guess what it made me wonder is, given what we know, at least from electrophysiology, of cross-modal plasticity in blinded or deafened rodents, for example. We know on an electrophysiological level of recording from neurons and cortex that there are significant shifts in the way that they function and process information. And given that we know that a species like a rat can also live in New York City or many other environments around the world, and similarly can adapt to niches, are you imagining that this kind of cortical plasticity that you see in humans really is human-specific or human-enhanced? Or do you expect that it's going to be more general, say, mammalian phenomenon?
That's a really interesting question that you're asking, and I like your example of the rat in New York, which is a good response to my claim. I guess I would say that probably the experience of a rat in New York is more similar to the experience of a rat in the forest than a person living in closer to nature and a person living near a city. But anyway, I think your point is well taken. Yeah, there's some empirical answers to your questions with regard to animals and cross-modal plasticity, and there's then some like theoretical ones that are hard to answer. So, there are of course people who write about this question of whether the human brain is especially plastic. My personal view, based on the kinds of data that we see and the reading that I've done, is the beautiful thing about humans that distinguishes them from rats is that they have this combination of specificity and plasticity. So these two things have often been kind of counterposed in the literature. The idea being we're either this domain general learning machine that can learn anything, or a collection of domain specific machines that can only learn particular things. And what I would say is that we have both of those. We have a collection of domain-specific machines and some domain general ones. They're both very important to the way we are as humans, but also that the domain-specific machines, at least early in development, are really amazing learners and flexible themselves, despite the fact that they're built to do certain things. So that's my general take on it. The other more particular thing to say is that it's interesting, comparing cross-modal plasticity in the rat and the human. is interesting. So I think the things that are likely to be the most analogous are cellular circuit kinds of mechanisms, and we can only study those in rodents. And I think that's really important. And there's a lot of beautiful, work by Takao Hench and other people doing like gorgeous things, understanding the underlying molecular mechanisms of plasticity. Exactly what functions take over the visual cortex in cross-modal plasticity are likely to differ vastly for a number of reasons. First of all, these animals have a much smaller brain, so their visual cortex and their auditory cortex are actually not very far from each other, unlike ours. Also, they don't have as much higher cognitive cortex to start with, so I think it's probably a human-specific phenomenon, though, when you have sensory loss, or at least it could be an enhanced phenomenon in humans and primates, that when you lose total input to some sensory system, there's considerable takeover by higher cognitive systems. So that's a long answer, so I'll stop.
Can I have a little follow-up? Yeah. So I thought, thank you for that was an interesting and good answer. And to get to this issue of equipotentiality, and to John's concern about when you have a new function emerging, is it because the function was there a little bit and only got sort of dialed up later? I think it's worthwhile sometimes to think about this in cellular and molecular terms, not just in functional terms. In other words, consistent with your finding that things are more plastic in humans early in life than they are later in life, axons can grow more early in life than they can later in life. So I don't think we should be thinking only in terms that the only kind of plasticity we can have is utilizing information for which there was already an axon running from place A in the cortex to place B in the cortex that can then be functionally turned up. I think we should I think that there is not equipotentiality, and I agree with you, and I think that there are constraints. But some of those constraints are not going to be about what was already there. They're going to be about what can sprout, what can grow. The other related thing that I'd like to say is that, we know that even among neuronal types like layer 5 pyramidal cells in the cortex, that in different parts of the brain, they can have very different electrophysiological functions, like very different maximal firing rates, very different rates of fatigue, very different bursting properties. And we can relate these to the expression of different ion channels. So I think it's going to be really fun to complement the kind of work that you do in model systems, to do something like you blind a mouse, and then you ask the question, what happens to the gene expression in a certain population of neurons in that area or in another area that is functionally changed in a blinded mouse, right? And we can ask that in terms of like what ion channel expression is turned up or down in a way that would support different kinds of firing and perhaps new computations to emerge.
I think that's super interesting, right? And you could have, and there's some work even in monkeys, I believe, looking at like which layers are primarily driving activity patterns in visual cortex. I'm now blanking on the authors and the details, but showing that the kind of feedback layers seem to be more influential in blinded animals, congenitally blinded animals. But I think that kind of work would be beautiful, because that would allow us to make the connection between what are the actual anatomical cellular changes that are possible if the experience happens early in life. I think that would be great. I think there are, with regard to humans, I do think we cannot rule out anatomical changes, and certainly there have to be anatomical changes that support the functional ones. The question is of what kind. Certainly, there are going to be microcircuit changes, synaptic changes, changes in like local neural communication. There could be even, lack of pruning. So you could imagine that in a human, and I've thought about this sometimes, but actually we have very little ability to measure them in people right now. Temporal lobe connections back into the visual cortex that are pruned in people who grow up with vision end up having less pruning. So you can imagine those kinds of changes, but I think in the human brain, it seems just less probable that there's like a long range connection, from the frontal lobe, let's say, or from the parietal lobe that was never there in an infant that grows. But I love this idea of looking at the relationship of anatomy and function.
Just one thing on sprouting, David. I've only looked at this in detail for supposed sprouting leading to functionally relevant new connections in stroke models. And basically it hasn't been shown. Okay, in other words, people see sprouting after injury, and you can get cross-callosal sprouting, you can get sprouting down into the striatum, you can see sprouting into other cortical regions, but no one, to my knowledge, has gone and taken one of those sprouts and cut it and shown a reversion of deficit.
Yeah, but these
So
are
in other
happening
words,
not in babies, they're happening in adults.
well, I'm saying, I'm just saying,
what Marina says, the plasticity is strong early and weak late.
right, but what I'm saying is that I don't think that sprouting is what's doing it, in so much that if you could sprout, you could have new regions do new things, rather than it happening exactly in the homologue, even in young.
Yeah, I don't think it does only happen exactly.
Well, I'd like to see the alternative data. That's what I'm saying. So, Melanie, you've been sitting there super, super patient. Shoot.
No, I'm really enjoying this discussion. I had a question about in your study on coding and the relationship with logic processing. You showed this overlap kind of bar graph with using this cosine similarity metric. And I wanted to understand exactly what that was and how you measure that, and also kind of how do you interpret it, and in terms of functionality or mechanism or anything like that. So just you can explain that.
Yeah, sure. Right. So I will give you a little bit of information about it, so to give you some details on how it was done. But basically, you can measure this overlap in a number of ways, and you get the same exact answer. And we've done it in a few ways. So if you just correlate, like you take all the vertices inside a particular area, like in the frontal lobe or in the parietal lobe or in the whole brain, and you correlate activity across one task with another, you can see the degree to which they're spatially similar. You can also look at the amount of overlap across two functions, literally like how many vertices are in common across them. All of these will give you the same kind of answer, which is that in this study in which actually it was done in two separate experiments, right? So in fact, what we're doing right now is designing an experiment where we have logic and coding in the same task. In this experiment, that wasn't the case because we had too many things that we were trying to pack in. So we had one experiment that localizes coding, which compares coding to its control condition, right, and various versions of coding. And then a separate experiment where participants do language, logic, and math. And those are all compared to each other. Again, like it would be nice to have them all compared to a control condition. And then what I'm showing you is literally a measure of the overlap between responses to logic versus language, like what parts of the brain are more active for logic versus language, and the responses to coding versus its control condition. So how many vertices overlap across those two contrasts versus across the contrast of language versus math or language versus logic. So with vertices that are responsive to language.
So I'm sorry, what is what do you mean by a vertex here?
Like a voxel. We're doing it on the cortical surface, so I'm calling it a vertex, but it's the same thing as a voxel.
Okay. And how, so this is a spatial correlation, right? Not a temporal correlation?
This is definitely, yes, this is evaluating the amount of overlap in space. Yes.
So
in cortical space. So like how many vertices are shared? Like
right.
To what degree are they activating the same parts of the brain?
And how do you interpret that? I mean, in terms of like, are these tasks using the same neural mechanisms in some sense, or are they just using the same areas,
Yeah, I think that's a great question. And so I think what we can tell from this is just at the resolution of fMRI, they're using the same areas, right? So this isn't really telling us the degree to which they line up like voxel by voxel. And actually, that's one of the things that we would like to look at, right? I think the way this comparison makes most sense is relative to other functions because they're all measured in kind of an analogous manner. So you measure language and math and logic in an analogous manner, and then you ask to what degree are the same brain areas involved across these different functions. And when you do that, you find that, although you might have thought that language would get reused for doing coding, it turns out that no, it's logical reasoning, the same brain areas that are involved in logical reasoning that get involved. But I think if we put these functions in the same experiment, which we hope to do, you can get more fine-grained kinds of measurements. And I don't know that I would expect them. Like one of the predictions might be actually that you do get some specialization for coding as differentiated from other kinds of logical reasoning as you gain coding expertise. Yeah.
And my understanding is you were looking at how at people interpreting code rather than actually writing code.
Yeah. So there are lots of possible things you could do, and we looked at code comprehension rather than writing code because it's a little bit, on the one hand, we want to do something that's rich enough that captures the phenomenon we're interested in this case, code. On the other hand, we're trying to do something that's controlled enough that will give us reliable responses. But yes, this is looking at comprehension, and I think we'll have to come up with some clever ways to study code generation. My prediction would be that similar mechanisms would be involved, but I think that remains to be tested. Yeah.
So do you think, Melanie, were you done? Did you want to ask?
Yeah, no, go ahead, John.
So on Melanie's point, I found that coding study really very interesting also, and I'm glad that Melanie brought it up. It's interesting, right, when you look at the dissociation between mathematical ability and linguistic ability, the work that Stan Dehaene's done, and then your work on coding, and then, work that Ev has done showing that a lot of cognition remains intact, even in global aphasics. Do you think that all of this suggests that the thinking that you can do when you're interpreting code, the thinking that you do when you're doing math, and even the thinking you do when you're dreaming up the perfect sentence to describe what just happened to you the other day, that it's cognition coming in various flavors that cannot all just be, as some people think, that language gets a lot of the credit for our ability to think at all. But it seems to me that your work on coding and work on math and then whatever's done, that really language is just one version of a tool that we can think with, and that we can think in a number of other ways. I was just wondering what your view of this was.
Yeah, I mean, I definitely agree with that statement. I agree with the idea that language is only one way of thinking. I also think that this evidence is consistent with the idea that, cortical systems, once they specialize for what they do, don't just get involved in anything willy-nilly. So there are some ways in which, coding stimuli share a lot of properties with language, and nevertheless, these language areas just don't care for them at all. And that's true also for mathematical equations. And that's the aphasia work, I think, is really important here. So yes, I think there's lots of ways of thinking. I'm actually very interested in this question of what is the relationship of language to thinking, and whether there are kinds of thinking that maybe don't share as many surface properties with language, but maybe nevertheless are connected to them. So anyway, I mean, I'm now working on a line of research to work with people who have differential timing in their access to language, because they're born without hearing, and unfortunately not all people who are born without hearing have access to natural sign language early in life. And there are questions to ask about how the language system changes in this population. but there are also questions to ask about different cognitive functions that people have done, like looking at theory of mind, reasoning, and numerical reasoning in people who don't have early access to
And do
a
they look
natural
good?
language.
I mean, are they pretty intact? In other words, do we know enough about that population young that to complement Ev's study in adult aphasics to suggest that they can, for example, become a grandmaster at chess potentially? Right? I mean, is that the direction potentially?
Yeah, I don't think the chess case has specific. I don't know of research on the chess case in particular. I think in general, the work certainly shows that, you know, people who don't have access to language nevertheless have a lot of remarkable reasoning capacities, just like all people do, that are intact. However, it's also worth saying that there's a big difference between being deprived of language early in life, so not actually having the benefit of language during development and during learning, versus losing your language as an adult. And you know, there's literature written by people who don't get access to language early in life. And first of all, it's emotionally and socially, it can be devastating. And for this reason, this is why it is so important for children who are deaf to have early access to a sign language in a way that blindness, congenital blindness, is not, which I think is like an interesting thing about humans, which is that language is access to language is fundamental, is just like fundamental to our being. There's also research suggesting that there are some elements of theory of mind reasoning, the development of which depend on, like, passing false belief tasks, access to language, in a way that, you know, although in an adult, if you have aphasia, you're perfectly fine doing the same kinds of theory of mind tasks. So this is work by Jenny Pyers and her colleagues. So, you know, those are subtle effects in some ways, and narrow effects. So they don't at all question the idea that there's a plethora of interesting, fundamental, uniquely human reasoning outside of language, but they do suggest that language plays a special and important role during development, including in learning and social domains, maybe other domains. And that's the kind of stuff that one of the kinds of things we're interested in.
Is logic uniquely human?
Yes, I do not have the expertise to really comment on this question. You know, and I think it depends on what kind of logic one has in mind. Certainly symbolic logic of the kind that we're using in our research study is uniquely human. Like if you say something like, if X and Y then Z, if Z then either X or Y, right? Like few people do that. There's gorgeous work by Nico Agnolotti showing that babies, human babies, do quite sophisticated logical reasoning based on visual events that involve, you know, no language whatsoever by 1 year of age. Now that's in humans, but it at least shows that there are important aspects of logical reasoning that are not dependent on language. That work is gorgeous, and I encourage everyone to take a look at it. I love it, and I hope to do follow-up research.
Could you post that in the chat, Marina? That you
Sure,
mentioned,
yeah,
you
let me
mentioned
find
Jane
it.
Pyer, and the person you mentioned,
Pyers, yeah.
and then this one. Could you just put, because they're so
Yeah.
fascinating? Could you just post those?
So I'm I'll let someone else talk while I find the.
Yeah, no, I mean, I was just wondering, you know, not whether babies can do logic, but whether non-human primates has anyone tried to teach them to do logic?
There's some work, which I think is I don't, you know, I really don't know enough about this. I know that people have done even work with like parrots, showing that they can do there's some controversy about exactly how much they can do. I see Chaz posted Nico's paper, which was lovely. So all I need to do is post Jenny Pyer's paper. Yeah, here it goes. This is a paper from 2009. Oops, no, I didn't. I thought I just closed our window. Sorry, someone else should talk while I get you the.
Yeah, I mean, to the degree that I've looked at some of that, Melanie, they're just, even compared to infants, they're just so bad, right? Fascinatingly, right? In other words, it's not even close.
Mm-hmm. John Rusky, hi. Do you want to ask your question while Marina is trying to find the link?
Yeah. Hi, Marina. Just
Hi
a fun
there.
fact, right before the pandemic, I was at Johns Hopkins, and I think Marina was probably one of the last people I saw before like the pandemic. Like it's one of the last trips I took that I remember. And I was like, oh my God, that's the last like academic trip I took. Very weird. Yeah.
How are
Anyway,
you doing? I hope you're doing well. It's nice to
I'm
see
in
you. I don't
France
want to
and
bore
feeling
everybody,
great.
but it's very nice to see you. Yeah.
Yeah. I love this. And I was thinking about the fact that, you know, as a linguist, it's interesting that no matter what type of sort of modality one expresses and understands language in, there are modality effects on the language. So it's like, you know, it's so common to think of like, you know, there's one language thing, and you know, blind people do language, and hearing people do language, and deafblind people do language. But there's all these little modality effects, right? So like sign language is not the same as spoken language, and tactile language is not the same as signed language, and not the same as spoken language. So it made me think like, if this sort of cognitive map shift idea that you had is sort of going, like what's the, you know, are there effects of like the say V1 or, you know, V1 or whatever, you know, are there effects of that that sort of impinge on the map that's being transferred, and like how do you tease that apart? I mean, I have no idea.
I think what you're bringing up is this beautiful point that we don't think about very often, I think, which is it's just remarkable the variety of ways that language can get into the mind and brain, right? So it's not, you know, there's sign language, which is visual. There's protactile ASL, which involves, you know, tactile sign on the body. There's Braille, right? There's visual reading. This is amazing. This is another way, I think, in which the human brain is clearly adaptable, right? Which is we have cortical systems that are equipped to do language processing. I think there's lots of good reason to think that humans evolved specialization to enable for things like grammar. But apparently, right, we have all this ability to adapt to getting it into our minds in a variety of ways. Now, and I think there's not, there's sometimes like this tension between understanding that all languages, and you know, linguists have studied sign languages, and of course Braille is just written version of English, right? The English Braille is anyway, and there's, you know, so whether it's a sign language or protactile ASL or a spoken language, there are key shared elements which are present in all languages, including, you know, like Russian and English and Chinese, right? So they all share fundamental properties of their grammar. At the same time, that doesn't contradict the fact that it would be interesting to understand the specific neural adaptations that enable humans to get language into the head in all these varied ways. And I'm certainly interested in that, including, for example, why is it possible for the tactile system to get language information, like through Braille, to language areas? What connectivity patterns is Braille making use of, which we certainly didn't evolve. We didn't evolve to read visually, and we certainly didn't evolve to read tactilely in particular. So there's clearly a bunch of pathways taking information to and from language areas, which we can then co-opt for the purpose of Braille reading and sign language. So I don't have any answers to the question that you're raising, but I think it's an underappreciated and beautiful property of language and of people's minds.
I think there was a Can I?
Oh,
I
sorry,
think
go
there
ahead.
was something in what you said, John, that I want to maybe formalize it in a different way. And John, tell me if this is one of the ways that you had it in mind, because I didn't see that reflected in Marina's answer, but maybe I misunderstood your question. So let me see if I understood your question. Was it that, let's say this is my cognitive map that or whatever it is that I'm using for language, and now I start learning Braille. So it needs to extend a little bit in some other area, right? And there is a way in which we can study how the brain extends this to include this other thing. So this is one way, and I'm an adult learning this. There is another way where I'm born, let's say congenially blind, I don't have anything. And if I had been seen in a possible world where I had been a sighted person, then this would be my map. But in a world that I'm not born like that, maybe it's like this because it's using some other regions. Now, what is the difference between these three cases that we discussed, which is the map, the representations, the kind of processes that the brain recruits in order to do language, which are necessary for it to do it, the kind of processes that it adapts to after having developed already, if it starts to learn Braille or ASL or something that is in a different modality, which is not as strange, to be honest. It's like it's vision. You just learn to translate some sense to another. So that's a little less, I would say that's a mild one. And then the strong case, which is that one develops from the beginning a map that recruits regions that, because one is not sighted, are doing something else than the other two cases that we were discussing, for instance, right? So it seems to me maybe there is a gradient of the differences between the representation / processes of a sighted healthy adult versus someone who acquired, who might be healthy or not, whatever different scales, acquired Braille or ASL, and then someone who was born without hearing or without sight and is recruiting some parts of the brain in a different way. So I thought that your question was beautiful in that it represented these different differences and potentially could have us finally be able to grasp what is the computational theory or claim in terms of what happens to these maps in these different cases. So what I understand Marina to say, I'm not entirely sure. So Marina, I'm going to offer a couple of things based on John's great example. One possibility is that the map really necessarily requires those visual areas in the congenially blind. The hypothesis of this one would be, this claim, the hypothesis would be if you sever the visual cortex, or let's say it's in a rodent, the example that David Linden was giving, and you have a mouse model of it or something like that, or rat model, then when you take away the visual cortex, whatever it is that the rat was doing that was recruiting this should be impaired. Or if it's a human, the human happens to thought experiment either with a lot of TMS or brain damage, loses visual cortex, they should show some impairment in that. Another hypothesis is it's not exactly necessary, but it expands that, which means maybe the congenially blind have some abilities that others might not have because they have extra cortex real estate that they're using for some processes. So maybe they will have higher verbal memory, or maybe they will be able to manage longer sentences or something like that. So that would be a similar hypothesis, but less strong than the first one. So it says it's not necessary per se. They're not going to get impaired if they lose the visual cortex, but they might actually be able to do it better than me, who is sighted, and I'm using my visual cortex for other things. I think there are some studies also showing that people who are not neuro typical, they use visual areas when they're solving certain problems also. So it might be the case that, and that's why they get, when they get sensory kind of overload, it's more difficult for them to process cognition. So similarly, I wonder the converse is the case with the congenially blind. If they are using that extra real estate, can their linguistic abilities exceed mine because they have this extra real estate to use that I can't use? Given we are using a scenario that doesn't require vision, so it's on equal footing to some extent.
Yeah, so that's a really good question. So there is some evidence. So there's good evidence actually that people who are blind have superior verbal memory. Yeah, so other people have shown this, and we found in a recent study that Karen Arkos actually did this work with me. What we found was that it's particular to verbal memory rather than like memory for sounds, for example, the advantage that you observe in blindness. We also found Rita Loyotili did this work with Connor Lane, showing that people who are blind are actually better at parsing garden path sentences, and to some degree sentences in general, particularly grammatically complex sentences. We're trying to, that's one study. We'd like to do it again. It's hard work to do. So I think there is some evidence. There's not evidence at the moment directly linking that to the visual cortex. And actually, you could imagine that people who are blind rely on memory and language as important sources of information. So we can't be certain that the effects are due to visual cortex plasticity per se. That's why those TMS studies are so important. So to your other question with regard to, I think a lot of possibilities are still on the table. So I will go on the record predicting that if you impair classic language areas in a blind person, that they will be more resilient to that than a person who is sighted, because they have this extra territory, because it's doing something for them. Based on the available evidence, I would also predict that a large lesion to the visual cortex would interfere with language function in a blind person. However, I think that that's a strong prediction, right? We'll see. It's an open question. I would also say that my current guess is that people who are blind have a more distributed language system. So it's not, so they're also using their prefrontal cortex, and they're also using their temporal lobe, and they're adding to it this occipital area. Although blind individuals are better on some of these language related tasks, they're not behaving like superhuman. They're not like Wonder Woman and Spider-Man put together as far as language works. So there doesn't seem to be like a proportional relationship between the amount of brain territory you recruit for a function and how good you are at it. So that leads me to guess that there's something more, that what blind people have is a more distributed system. That's my current guess, but I think actually there are a lot of possibilities on the table, including one where blind people have a more distributed system. There's a bit of an advantage that comes from having this extracortical territory, but it also is necessary. And the cost is that if you then damage that cortical territory, then you will impair the function. So I think there's a lot of possible worlds still on the table that remain to be distinguished between.
I can just say to blind patients getting stroke, right? I've seen a bunch of blind patients who, when they have classical lesions for stroke, are just as aphasic as the non-blind patients are. So, in other words, they don't seem to be relatively, not that I've studied it, but I've actually commented.
You were talking about you have congenitally blind patients who have lesions to frontotemporal areas. Why aren't?
I don't remember if they were congenitally blind, but I don't actually remember. But I've had, in the course of my lifetime, some blind patients who've had strokes who have become aphasic, right? And I do not recall them being particularly resistant to the lesion. I don't know how many such patients there would be, but I think it's to the point that Ida's been asking throughout that I would be, it's a strong prediction, right, Marina, that you could have
It's a prediction for congenitally blind people, just to be clear, and only for congenitally blind people, because the fMRI data suggests that this phenomena just does not happen.
Right, they can't do it for whatever reason, even though they have, yeah.
Would you predict the same thing regardless of modality? So the same thing of congenitally deaf people, congenitally deaf-blind, say for auditory cortex, STS, et cetera, et cetera? Or
that's like a great question. That's a great question. And I have we're starting to work up in this area now. That's what we're gearing up to do. And I have a collaborative project now with Marcin Swed and his student, Maria, trying to address this question and people who are deaf. I think that it's an open question, right? So the visual system and the auditory system are very, very different. The auditory system is just a smaller, less developed part of the brain. It has a different connectivity profile. So I don't, and actually, like, while in the case of blindness, there's lots of evidence that V1 takes on other functional responses. There's less evidence of, there's some evidence of effects like that in A1, but actually the evidence is kind of mixed. So I think the jury's open. My first prediction would be that this invasion of sensory areas by higher cognitive systems in cases of total sensory loss is probably a general phenomenon. So that would be my first guess, having to do with the fact that our brains are mostly higher cognitive stuff, and there's very few connections among sensory areas in humans. So that would be my first guess. But like exactly in which ways the plasticity is similar or different, this relates also to some of the things that John was saying. The phenomenon could very well be quite different in the case of the motor and sensory motor system. I just don't know. Yeah.
So just to bring back to the two things that I was mentioning, both are about lesions in the congenitally blind, in the visual cortex. The question is, will the language deficiency that you're predicting, is it that the language advantage they have will diminish? Or is it that their core baseline language ability will diminish compared to a sighted adult?
I think that's a great question, and I don't know the answer to it. I think my first guess would be that the language capacity will actually diminish. That's my first prediction, but I just don't know. I mean, because my guess would be once you add a cortical area to a network, you can't just cut that area out in adulthood and expect to have no effect. Even if during development you have the opportunity of building a system that didn't involve it. That's my first guess. But, it's an open question.
I think that's a fair comment, that if something since development got involved, it might not. But I want to give an example in healthy adults, mice, rodents, humans, mammals in general. And the example that I will give, again, it's an analogy, so please don't think that I'm drawing a one-on-one mapping. The example I'm trying to use is one where we observe that the entorhinal cortex also shows place fields, for instance. However, when you study it, you realize that these place fields form after the hippocampus forms the place fields. If you take out the hippocampus, the entorhinal cortex, I'm mentioning a wrong region, actually. It's right here. These cortical place fields that are not just in entorhinal. Actually, there's like a lot of other parts of the parietal cortex have some kind of same gradient even of different place fields of different size that is comparable to the gradients in the hippocampus. However, if you lesion the hippocampus, that thing will not get formed. But if you lesion those regions that are in the cortex, the hippocampus can still do its place field work. Other things might get impaired, but the main thing that the hippocampus does with the place fields and like the spatial navigation, that thing won't get impaired. Now, if you impair the hippocampus, parts of the brain, the maps that I've very much overlaid in the past and I have completely consolidated in my brain, I can still navigate those maps. I can still find those old addresses. But if you change my home address, I cannot find my new home. I cannot learn a new map because that would require for. So there are ways in which even regions that since birth are being sharing information or showing information in them in the cortex that was that in some part of the cortex that actually originates elsewhere in the brain. The fact is that the originator still is the one that's generating it. The other place uses it, broadcasts it, makes it easier, available to other places, helps consolidation, etc. But it's not the generator of it. And so if you do a lesion, the one that really disrupts the situation and is really necessary is the one that generates. So I think this is, again, another way of asking the same kind of questions, but using those two models, I think you made it clear that your assumption is towards the stronger model as opposed to the weaker model. And I wonder how you would compare that suggestion with this observation, that if you have a region that since birth even reflects information that's generated in another area, if you take it out, that region is not impaired in generating that area. So I wonder whether this is somewhat similar in the sense that they might be using it for many things to improve language, but if you take it out versus take out the frontal language regions or, you know, Broca and Wernicke's area, that might be a different kind of deficit. So yeah, that's my question to you. I hope that was clear.
Yeah, I think what you're asking is an empirical question to which we don't know the answer. The only evidence we have that I'm aware of is one study. So, you know, and I keep repeating it and kind of feel silly doing it because it's just only the one we have. There's also some evidence that you can impair Braille reading by TMSing V1 in people who are blind. A couple of studies showing that. So I think we know that. But then there's the study showing that if you can, that people who are blind are more resilient to frontal lobe stimulation when generating verbs, and show errors when generating verbs when stimulated in the visual cortex. This is the Mirametti study. Now, you know, this again, TMS is also different from a lesion in the sense that maybe given time to adapt to a lesion, the effects would be different. So they're not exactly analogous. I really just don't know the answer. My prediction, I prefer to make strong predictions rather than weak ones. I'd rather be proven wrong. But my prediction based on the evidence that I have is that lesioning, a large lesion to the visual cortex would produce an aphasia-like impairment. Let's find out in a person who is born blind. But what I do think there's some analogy between the case that you're talking about, if I understood it correctly, and I like to read the paper so that I can, like, say, be more informed. In the case of development, which is to say, it is very likely, given the other various kinds of evidence that we have, that there's some neural predispositions present in language networks of human babies that enable them to do language and enable the baby to acquire language, and then in a baby who's blind, enable it to colonize the visual cortex. So I think there's a developmental relationship, there may well be one, such that if you weren't born with a Broca's or Wernicke's area, then you could never develop those functions in the visual cortex because they are like the seed, the germ seed that is required to begin the process. That's my current take on it in the developmental progression. So in that way, there is a primacy to these areas relative to the visual cortex area. But then in terms of an adult function, my prediction is that there's no primacy. The other thing is that, you know, exactly there's a lot of change that happens in language areas themselves during development in an infant between when they're born and when they actually acquire language. And so what those areas start out with, like, they have some innate capacity to develop into language areas, and the place where they end up and their size and their functionality and like what other areas they recruit may well be quite different.
Marina, I was going to ask, this was the second question that I was going to ask you, was sort of a flip side, and you almost just brought it up right now. And I was wondering, like, within a modality, say speech, or, you know, speech and spoken and hearing abled people, it's pretty, like, things are pretty stable. You know, like between people, I mean, there's a little bit of variability, but by and large, things are pretty stable. So that seems pretty weird from this perspective of, you know, if the brain wants experience dependent, it can just map stuff wherever. So, like, you know, it seems like it's a very weird thing to expect on a population level, you know, much less even in one community, say, that, say, the same brain area does the same thing for the, you know, wildly different people with wildly different experiences and linguistic input and whatnot. So, like, why should that be the case?
I'm going to one-up you and
better.
say that there's consistency across blind people, right? So it's the visual cortices are plastic, but across people, there's some similarity in which parts of visual cortex end up, for example, responding to language, and which parts of the visual cortex end up responding to math versus executive control. That's kind of crazy, right? So the way I think about it is, you know, we have strong anatomical predispositions, both connectivity wise and cytoarchitecture wise. The connectivity in particular, the reason I keep harping on it is the long range connectivity is present at birth and determines the flow of information. So why do I think these areas end up in the visual cortex taking on these functional profiles? Because they have biased connectivity. The functional connectivity data supports this idea with language networks and, let's say, number networks in this case.
But Marina, I mean, the thing is that's the opposite of reorganization. It's my argument that it's already preordained to have that connectivity that has the potential to do what it does. I don't understand why you're not just saying it. I mean, reorganization implies an ability to transcend your previous identity. But if you're going to,
I think there is both. That's my argument, which is that specialization and the ability to transcend your original function fundamentally, in the most profound way possible, are not contradictory. So what I'm arguing is that humans are both specialized and have a lot of structure built in and a lot of systematicity, but that the functional character of a cortical map, what the function is, depends partly on the input it receives during development and what other areas it's interacting with. So that is, those two things are.
It seems to me, for me, it seems that there's going to have to be a much clearer conceptual apparatus to make that distinction. I just don't think that they sit as easily together as you're saying. In other words, if there's some jitter that's possible on top of the connectivity, I agree with you about that.
I don't know what you mean by that.
And I tried to,
I don't get it.
And I try to clarify this. Specialization versus domain specificity versus quality has been very often discussed in the domain of the prefrontal cortex. There are some people who every now and then write a paper and they're like, it is domain general because we found it active in this and this and that. And then some people say, oh no, it's domain specific because look at these very tiny differences we found between analogical reasoning and social inference. And then the other ones come and say, oh no, so this discussion obviously has been going on. The cortex is not the same in the entire prefrontal cortex. There's cytoarchitectonic differences. Their connectivity with other regions is different. Their interconnectivity within it is different. I'm very much on the side of the specialization. And I think that the reason for that specialization, even though there is some flexibility, is not completely flexible. In other words, it's not a randomly connected network that, depending on how it develops, it would specialize in different ways. So I don't believe that. And I think if you're saying that there is specialization in the brain, it seems like you're saying it's kind of random and then the specialization depends on development. I think so that's one possibility. Maybe you're not saying that. Another possibility is to say there is specialization, but if you change something in development, the specialization can change a little bit, but there is limits to it. It doesn't completely change. Not every region can do everything. Some regions can get recruited depending on their connectivity with other regions that are doing something already. Or you can go even more extreme and say, you know, there is some differences. Some regions might kind of get appended to other regions, but really they're still doing some functions that are similar to whatever information they're receiving from the connectivity due to synchronization, but it's not fundamentally different. So the first one, this is a spectrum that I provided for us to think about. One says it's all random and specialization that we observed is just contingent upon how you would connect things to each other. Another one says, no, it's not entirely random. There is some structure and some of the layer differences and in connectivity differences. And by that, I don't mean just functional connectivity, but also just structural connectivity differences are part of the reason that there are limits to what can specialize how. So you can get some differences if you change development, but it's not free for all. It's not that anything can do anything. It's not that visual cortex can do anything. And it's not that it kind of so that's the question. To what extent can it take over? Is there a limit? And there is one that is more restrictive that I think maybe John is in a more restrictive domain. I think I'm somewhere in that middle where I think there is a limit and there is a calculated and measurable limit to what each of the cortical regions do, depending on many features of them that makes them distinct. And it's not entirely random, and not everything can do everything. But I wonder where you stand on this spectrum. Clearly not at the John end of it, but between the middle and the beginning, the random and everything can do anything in the cortex, I wonder where you stand there.
Yeah, so I guess what I'm trying to argue is it's not really, for me, a continuum between like anything goes and just perfect, it only does this thing. So for me, it's a dissociation between anatomical specialization and functional flexibility. So what I think is that the human brain has a lot of anatomical specialization. It's very ordered, very particular cytoarchitecture, very particular anatomical connectivity, but that in particular in the case of the cytoarchitecture, the same cytoarchitecture is compatible with representing drastically different kinds of information. So we cannot map from content, from the level of analysis that describes cognitive content to anatomy in a straightforward way. Another piece of data, just before I move on, that just like is worth thinking about is Morgan Kassur's studies from 30 years ago now, where he rewired the ferret thalamus to project, what was it now? I always get this mixed up, to project visual information
to the auditory,
into the auditory,
to
auditory cortices.
But even
right?
that was very limited, and it relied on a topographic A1 V1. It wouldn't have worked if you'd stuck it into prefrontal cortex.
Well, you don't know that, right?
Well,
so
yeah.
The experiment hasn't been done. That's just like, okay, you think it wouldn't work, and I think it would work. So there you are, and what if you just added a chip to your cortex?
So that would be, that chip is a different kind of cortex now. So that chip is a different part of the brain now. Let's keep it to the conceptual question, maybe. I want to make sure we have a focused conclusion to this in terms of commitments.
Sounds good. And actually, I'm so sorry, but I really, in like 2 minutes, I should run it.
We went
I've
much longer.
abandoned
We
my
apologize.
family.
So we do a very rapid kind of question and answer, so it doesn't take long to answer smaller questions. So is there a limit, and just a yes or no, to, let's say, visual cortex doing functions that other parts of the brain do, doing them, not just showing information, not just co-activating, not just synchronizing, actually doing those functions? Is there a limit to it? Or can it do anything any other part of the brain can do? Let's say cortex. I guess, can it?
You're asking a question to which the empirical evidence doesn't currently hold the answer. So what I can tell you is, based on the data from the blind participants and the blind population, there isn't a limit to the kinds of functional responses that we measure in neuroscience that the visual cortex can assume, the kind of information that these neurons can be driven by. I would argue strongly, based on the blind data, there isn't a limit. The data with regard to the question of functional relevance and behavior, I think there's a little bit of evidence, but the data just aren't there to clearly say one way or the other whether these visual areas are supporting these higher level cognitive functions. And I hope we get those data, but they just aren't there. So I don't know the answer to your question.
So the reason I asked the short answer is because it's not an empirical question based on evidence. I absolutely don't think you have evidence to answer this. I don't think that fMRI studies are sufficient to answer this. The question was a hypothetical one to figure out your theoretical commitment. So I want to understand. So you think there is no limit. So the specialization is just contingent upon the development, but anything in the brain can do, in the cortex can do anything else. I guess the answer I got from you now.
No, not really.
Okay.
I mean,
Okay.
no,
So
in the sense that, but I think it's much more complicated than just kind of putting it that way, right? So this is what I was trying to say with regard to the language areas and their innate function, right? Mm-hmm. So what I think is, there's some innate predispositions that enable the human brain and not a chimp brain to do language, right? There's something innate that's particular to human brains, but I think that the adult state is a product of both those innate predispositions and the learning that happens during development. And during that developmental process, a lot of functional change can happen and areas can assume functions they never evolved for and didn't have at birth. So yeah, I don't know.
I think that on that spectrum, you're not entirely at the extreme. I think I'm more in line with that framework, that theoretical framework, because chimps and humans have very similar visual cortices, but definitely not as similar prefrontal cortices. In fact, the entire area I work in, it's some parts of it don't have a homologue in other species in the PFC environment area. Some parts of it does, some parts of it don't. So I think it's very interesting that you're mentioning, and I am with you on that. I think we might differ on how far that particular generativeness of those circuits can be accommodated by other regions or not. I think we might differ slightly, and that would be an empirical thing. But I do think computationally, now I have a better idea of the theoretical commitments that you have.
So what I would say,
go on.
Well, I would say, Marina, of course, this has been absolutely fascinating. I think what's really meta-fascinating to me, and I don't know what you think about this, is that for something that is so fundamental to neuroscience, and as you said, the Manganko-Sur experiments, fascinating experiments with TMS over the occipital cortex in Braille, amputees, congenital hemispherectomy, cases, that we're still very much in the conceptual, we're not really resolved. Wouldn't you agree that, in other words, that you would have thought that we'd be further along in terms of it, whether it's a spectrum, like Ida says, skill learning in a healthy brain, adapting to New York when you should have been on the savannah in a healthy brain, to having an amputation, to having a congenital stroke, that this spectrum of change, reorganization, and plasticity is still very much up for grabs, right? In other words, would you agree that it's not resolved as much as some people might think it is, or are we just being difficult and you think it is more resolved? Do you know what I mean? It just seems like it's still a nexus for deep investigation and disagreement. Is that true?
Yeah, I certainly agree with the idea that it's a nexus of deep disagreement and argument. And yeah, I think, and there's a lot of reasons for that. One is research is hard. And another reason is that I think the answers, fortunately for us, turned out to be much more interesting than the questions. So in other words, like the picture is much more complex than the original questions that we were asking, way back. So I think we have made a lot of progress. Really, we have. If you just go back to 40 years ago, I met a woman who had a daughter who was congenitally blind, who was told by her doctor that her visual cortex would atrophy, right? Like, we've known that that's not true for a long time. And that was up for debate for a long time. And just even that these areas are responding to other kinds of information but we're still trying to work it out. So yes, I think there are a lot of pretty different ideas that are still available. And it turns out that I think there's also a lot more plurality in the way that the brain does and doesn't adapt, depending on the cortical system and the domain. And anyway, it's so much fun.
So there isn't going to be, would you agree then, there won't be a universal theory of cortical reorganization or plasticity, right? It seems like there's going to be, we're not going to have a final theory of cortex which just gets modulated by how soon after birth it happens. It's not going to be as simple as that. We're going to have to be domain specific, cortical area specific, lesion type specific. In other words, we're not going to have a universal theory of cortical plasticity.
Yeah, I'm not quite that pessimistic. I think we will discover the principles. And once we discover the principles, we will be able to make predictions for each system. Mm-hmm. But I think that the systems are different enough that when we apply the principles, they will interact in complicated ways. Mm-hmm. So I do think that there will be a unified theory, but I don't think it will be like straightforward to say like, oh, this area is going to change or not change. It's going to be more principles of anatomy and adaptability and function that we will ultimately understand and be able to know. Yeah.
So I want to say, yeah, given the time and everything, I first want to say,
thank you so much,
Marina. We're a little extreme in discussion here, but
great. That's great.
Really, I learned a lot.
Get to the bottom of it.
I'm sure John and everybody in the audience feels the same. We're a little rusty. It's like the first session of the year. John is in a different time zone. So
And I have COVID.
And you have COVID. So if you were a little too excited, you understand why. It's really out of passion. And I think that's one of the things I love about these discussions. It's a little bit like being with friends around the dinner table and really just authentically asking questions, showing emotions in your face, etc. So I really appreciate that you were patient with us, that you really participated in the discussion, because I feel like the discussion was a lot more clear for me. And I feel like now I realize there is this whole interesting field for theorizing computationally that is very easy to make simple models and write like a nice opinion piece by yourself, maybe in a way that you can clarify those commitments and predictions. And I loved what you said in the middle of your responses, which was that you prefer to make strong predictions because when they are proven right or wrong, we learn more from them. And I really appreciated that you said that. So although initially I thought, oh no, these predictions are too strong based on the data, then I realized, oh, your philosophy of science is to over predict on the basis of the data, because then your theory can get more or less information when you are proven right or wrong when you do the experiment. So I appreciate that you explained your philosophy of science to us. And I think it made me realize that some frustrations I had earlier on was because I was operating from a different philosophy of science. And I really respect that. Thank you so much. I hope that everybody appreciated this. Feel free to read the comments later if you wanted to, the questions that we didn't get to ask. It's all in the Ask a Question area whenever you want. And yeah, if you want to say outro to lead us out of today's session, please go ahead.
I wish I could have a drink with you guys. It's time for a glass of wine together.
It is.
That's
My glass
what I
is
want to do
fitted.
now.
And for everybody who's still in the room,
Virtual.
Yuri Buzaki will be with us in 2 weeks. Is that correct?
Yes, that's correct.
Thank you.
Thank
Take
you
care,
very
everyone.
much. So much fun.
Care.
Bye.
Bye-bye.
Thank you. Bye-bye. Enjoy your dinner.